All right, my lovelies. Week 1, day 2. I know I'm behind. Imagine that. So we're going to be on page 80 to page 88 for all of this. This one is entitled Muscle Power. Gene therapy builds up muscles to fight AIDS virus. Muscles defy gravity. They resist the planet's downward pull, enabling us to stand upright and move. Now, a team of California researchers has endowed muscles with a new kind of strength, the power to combat diseases. The researchers injected muscle tissue with tiny pieces of genetic material. That material transformed the muscles into factories of proteins that attacked viruses. Although the technique has been tested only in laboratory animals, it defends them against one of the deadliest of all infections, acquired immunodeficiency syndrome, or AIDS. Human trials could begin in 2013. About 7 years ago, but hey, whatever. Pseudoinfection. Since AIDS was first identified in 1981, it has killed more than 25 million people around the world, nearly 30 million according to www.unaids.org. AIDS disarms the immune system, leaving the body without protection against disease. Medical researchers have devised several drugs that keep people from getting sick after they've been infected with the human immunodeficiency virus, HIV, which causes AIDS. But no one has yet come up with an effective HIV vaccine, or a drug that guards people against becoming infected in the first place. Some HIV vaccines exist, but even the best one shields only 3 out of 10 people from infection. A vaccine is a safe germ, a normally harmful virus or bacterium that has been disabled or killed in the laboratory. Though the germ can no longer cause disease, when injected into the body, it still promotes the immune system to create specially designed proteins called antibodies that destroy it. The immune system remembers that response and will mount it again if the body ever comes in contact with the harmful germ. Effective vaccines have been designed to prevent many infections, among them mumps, measles, hepatitis, polio, various flus, and whooping cough. HIV vaccines do not work well, it appears, because HIV targets the very defense mechanisms of the immune system that protect us from disease. A lucky few individuals are naturally resistant to HIV. Their immune system manufactured powerful antibodies that destroy HIV. Those antibodies are called broadly neutralizing antibodies. They are a key to a new treatment devised by scientists at the California Institute of Technology. David Baltimore, a Nobel Prize-winning medical researcher, oversees the lab where the work is being done. Scientists have known about the anti-AIDS BNAbs for some time. They've also located the genes responsible for the BNAbs. A gene is a segment of DNA that instructs the body to make certain proteins. Mighty mice. The Caltech scientists have used the BNAb genes on a group of specially bred mice. Most mice don't develop AIDS. HIV has no effect on them. But the Caltech mice are vulnerable to HIV. They react to it the way people do. The first step in the Caltech experiment involved endowing the mice with the BNAb genes. That was done by inserting the genes into the harmless virus, then injecting the virus into the leg muscles of each mouse. The virus then slipped the gene into the mouse's muscle tissue. The mouse's muscles, not its immune system, started making the antibodies. Step 2 involved infecting the mice with HIV and then monitoring their condition. Would they get sick, or would the antibodies produced by the BNAb genes protect them? The researchers began with a tiny dose of HIV. The mice showed no signs of sickness. The dose was increased, still no symptoms. Even when megadoses of HIV were given to the mice, the animals remained in the pink of health. We expected that at some dose, the antibodies would fail to protect the mice, says Alejandro Balaz, a member of the Caltech team. But there was no infection, even when we gave mice 100 times more HIV than would be needed to infect 7 out of 8 mice. Baltimore is very encouraged by the results. We're not promising that we've actually solved the human problem, he says, but the evidence for prevention is there in these mice is very clear. The logical next step, says Balazs, is an experiment with human volunteers. We are currently in discussion to start a clinical trial for this approach in people and are at the early stages of identifying exactly how this will happen, he says. It will take at least a year or more until we could start. Risks and rewards. HIV research is risky work. We have special procedures that we follow, explains Balazs. This includes special protective clothing like gloves and a face shield to prevent splashing. We have special rules about handling sharp objects when working with the virus. We also make sure to sterilize every surface that it comes into contact with. Still, it's work with an outcome that couldn't be more valuable. I've always been interested in how the immune system keeps us safe from all the diseases that are out there. I was especially interested in HIV because it is a disease that causes some of the worst suffering around the world. Balazs says, when I heard about the idea of using other people's antibodies to prevent infection, it sounded like a really promising way to approach the problem. HIV infection. HIV, the virus that causes AIDS, disables the immune system. It invades immune cells called T helper cells and reprograms them to become HIV factories. The number of healthy T helper cells gradually dwindles, and the body becomes defenseless against diseases such as pneumonia, tuberculosis, and lymphoma. And there's a little graphic here that you'll want to take a look at. All right, that's all for your reading for today. See you later.